So this really brings us to the importance of T cells in the response to SARS-CoV-2 infection, which is what I'm going to focus on in the next couple of slides. So the first point that I want to make here is that SARS-CoV-2 specific T cells can be detected in seronegative individuals. So as we've discussed, some individuals who suffer from mild or asymptomatic disease might not test positive using an antibody test, but many of them do appear to have T cell responses. And this graph comes from a publication which was produced by the Karolinska Institute, which looked at T cell responses versus antibody responses in healthy blood donors, exposed individuals, and people who suffered from mild or severe COVID-19 disease. And what was really seen was that the T cell responses were consistently higher in these populations than the antibody responses. And what the publication showed was that almost twice as many healthy individuals who donated blood during the pandemic had memory T cells versus antibody responses. There was then another paper that came out around a similar time, which came from a French group, which showed that SARS-CoV-2 exposed family members demonstrated SARS-CoV-2 specific T cell responses, sometimes in the absence of antibody responses. And both of these papers really concluded that if we use seroprevalence as the sole indicator of whether someone's been exposed to SARS-CoV-2, it might lead to us really underestimating the extent of population level exposure or potential immunity to SARS-CoV-2 infection. So what this really means is that T cell responses might actually be a more sensitive indicator of whether someone's been exposed to the SARS-CoV-2 virus than antibodies. More really reassuring data has come out around T cell responses in SARS-CoV-2 infection. And some of this data is really starting to suggest that T cell responses might be really robust and really long lasting. So far in the pandemic, T cell responses have been detected in samples that have been taken from patients who've recovered from SARS-CoV-2. And this data comes from a publication which came from Alessandro Setti's group. And it showed that 1 month after infection, or 1 month after the onset of symptoms, 61% of patients in this study had detectable circulating memory CD8 T cells. And when we look at the patient population 6 months after infection, you can see that around 50% of patients still had those detectable CD8 T cell responses at that time point of 6 months after the onset of symptoms. So what this tells us is that SARS-CoV-2 specific T cell immunity can be maintained at 6 months following primary infection. And there's now additional publications that are suggesting that this T cell immunity may be extended up to 9 months post-infection. And what this tells us is that SARS-CoV-2 specific T cell responses might actually be able to outlast those transient short-lived antibody responses that have been shown in other publications. So all of this really leads us to the question, could T cells be a good adjunct test to antibody testing for SARS-CoV-2 infection? And this schematic is exactly the same as the one that I showed earlier, but this time I've added on the T cell mediated response so that we can see how that fits into this picture. And what it really shows is that cell-mediated immunity or T cell testing might be a more sensitive way to detect past exposure to SARS-CoV-2. And there are a couple of reasons for this. And the first one is that T cell-mediated immune responses are generally generated prior to antibody-mediated responses, which is why we see this red line coming up first. And the reason for this is that we need those SARS-CoV-2 specific T cells in order to prime the B cells to produce the SARS-CoV-2 specific antibodies. So we do generally see those T cell responses prior to antibody responses in the immune response. Another factor is that some patients may be able to eliminate the virus using just innate immunity and T cell responses, and they never actually have to generate those antibody responses, and as such, they never seroconvert. And finally, recent publications have shown that the SARS-CoV-2 antibody response might decline over time, but more publications are showing that T cell responses might be really robust and really long-lasting. So now I'm going to change track slightly and talk about the principles of cell-mediated immunity testing for SARS-CoV-2 infection. So the real basics of this are that someone becomes infected with SARS-CoV-2 and their T cells are exposed to SARS-CoV-2 and they become activated and develop into SARS-CoV-2 specific T cells, and that's both CD4 and CD8 subsets. What this means in the body is that SARS-CoV-2 specific effector T cells can migrate to the site of the infection where they start to secrete a cytokine called interferon gamma. And this interferon gamma recruits immune cells to the site of the infection and activates more immune cells called macrophages to attack the virus. Now, what interferon gamma release assays do is take advantage of this in vitro to assess the immune response to the virus. So infected patients have SARS-CoV-2 specific T cells circulating in their peripheral blood. And when these T cells are stimulated by SARS-CoV-2 specific antigens in vitro, they will start to secrete interferon gamma. So interferon gamma release assays use this interferon gamma secretion as an indirect measurement of whether someone has been infected with SARS-CoV-2. So the idea here is that any T cell that has seen or come into contact with the SARS-CoV-2 virus in the past will produce interferon gamma in vitro when they're stimulated with SARS-CoV-2 specific antigens. And this is the real basis of using interferon gamma release assay technology to test for exposure to SARS-CoV-2. So now I'm going to move on and talk about the Tspot Discovery SARS-CoV-2 Research Use Only Kit. So the Research Use Only Kit is based on the Tspot technology platform. And the Tspot technology is a proven technology which has been approved for clinical use to detect TB in over 60 countries. The platform uses a standardized sample prepared from peripheral blood, and it allows the number of responding T cells to be enumerated for a really precise assessment of the T cell response. And this standardization means that the test is able to maintain performance even in samples from immunosuppressed individuals who may have reduced T cell counts. There is also an automated platform which offers 54 hours blood stability when the test is performed with the T cell select reagent kit. Now I'll talk a little bit about how the Tspot Discovery SARS-CoV-2 Research Use Only Kit works. So it starts with a sample of whole blood, which is taken by a routine blood draw. And then from this whole blood, we isolate out the peripheral blood mononuclear cells, or the PBMCs. And these PBMCs are roughly 70 to 85% T cells. So it's really enriched for the population of cells that we want to study. And once we have these isolated PBMCs, the cells are washed in order to remove any interfering substances like toxins or endogenous cytokines that might interfere with the results of the assay. And then the washed cells are counted before they're plated into the assay. And this counting means that it really doesn't matter if there are variations in individual cell counts who are being tested, because there's the same number of cells going into the assay for every single individual. So once the cells are washed and counted, they're plated into the test. And this is where they're challenged with the SARS-CoV-2 specific antigens. So the idea here is that any cell that has come into contact with SARS-CoV-2 in the past will recognize those specific antigens used in the test and start to produce interferon gamma. And this interferon gamma is captured by interferon gamma antibodies that are coated on the bottom of the plate. The next step is to add a secondary labeled antibody, which binds to the captured interferon gamma on the bottom of the plate. And then finally, a detection reagent is added, which produces these bluey purpley spots on the bottom of the plate wherever interferon gamma has been produced. So what these blue purple spots really show is the footprint of an individual cell that was producing interferon gamma in response to the SARS-CoV-2 specific antigens used in the test. So we really get this single cell resolution result. So this slide really talks about the antigens that we use in the research use only test. And all of the antigens used in the kit are based on and span the length of the structural proteins of the novel SARS-CoV-2 coronavirus. And these proteins are the spike glycoprotein, and that's the S1 and S2 subunits, the nucleocapsid protein, membrane protein, envelope protein, and a mixture of structural proteins. And all of the peptides used are designed to elicit both CD4 and CD8 T cell responses, and any specific sequences that have high homology to endemic or non-SARS-CoV-2 coronavirus has been removed to improve the specificity of the test. And now I'm going to talk a little bit about the test interpretation. So every single patient will have a panel which is dedicated to a nil control and a positive control. And the nil control is just cell culture media to ensure that there's not a large non-specific interferon gamma response happening. And the positive control is PHA or phytohemagglutinin, which ensures that the cells being used in the test are functional, they're responsive, and they're alive. And then we have our 5 SARS-CoV-2 specific antigen panels. And these are really the panels that allow the user to count the number of SARS-CoV-2 specific T cells out of the 250,000 cells in each well that are responding to the various SARS-CoV-2 antigens used in the test.